Hi, my name is Taylor Christ, and I'm here to guide you through our third lab. Today we're going to be talking about connective tissue, which is a somewhat confusing subject at first, but you will get plenty of practice with it, as it is present in just about every slide you'll look at. Today we're really only talking about connective tissue, so please don't worry about trying to identify everything that's in the slides that we're looking at today. So connective tissue is unique in that it consists primarily of non-cellular matrix with some different connective tissue cell types intermixed among that matrix. And by the end of today's lab, you should be able to identify what kind of connective tissue cells are residents and what kind of connective tissue cells are wandering or transient cells. You should also be able to tell the difference between collagen fibers, reticular fibers, and elastic fibers, knowing what they look like in the histological section, and being familiar with their basic functions in vivo. And lastly, try to be able to explain the general function of the extracellular matrix components as a whole as it pertains to connective tissue. Let's get started by taking a look at slide 232, which is thick skin stained with H&E. The skin, which we're going to talk about in more detail in a later lab, is composed of three basic layers: the epidermis up top, the dermis in the middle, and the hypodermis seen here at the bottom. The cellular epidermis at the top is the most superficial layer and is composed of stratified epithelial cells. Just deep to the epidermis, the dermis is composed primarily of connective tissue, such as collagen, while the hypodermis, the deepest layer, is composed of fat and some loose connective tissue. So now that you're oriented, let's take a look at the dermis, which again is located between the epidermis here and the hypodermis down here. It itself is composed of two separate layers. The papillary layer, right under the epidermis, is more superficial and composed of loose connective tissue, while the deeper reticular layer is composed of dense connective tissue. The dermal papilla are these arches that extend superficially into the epidermis. At 10x, you can see that this region is more cellular, and the collagen fibers are less densely packed, as in the deeper reticular layer. Again, the papillary layer of the dermis is composed of loose connective tissue. The randomly patterned pinkish eosinophilic material seen here in the deeper reticular dermis is dense irregular connective tissue, also composed primarily of type 1 collagen fibers. Dense irregular connective tissue will also have some elastic fibers and reticular fibers that, remember, you can't see because they're not stained with H&E, that help to function to protect the body from stress and strain in all different directions. A transverse section of type 1 collagen can be seen here by the green arrow, and a longitudinal section of type 1 collagen can be seen right here next to the blue arrow. Alright, so we've moved to a different section of the slide to take a look at fibroblasts, which are seen here with their elongated nuclei. Fibroblasts are responsible for producing collagen and other components of the extracellular matrix of the dermis. Notice that there are more fibroblasts visible in the papillary layer than in the reticular layer of the dermis. And while we're talking about other components of connective tissue, I'd like to point out that there are two basic groups of connective tissue cells: resident connective tissue cells and transient connective tissue cells. The resident cells include fibroblasts, dendritic cells, mast cells, macrophages, stem cells, and adipose cells, while the transient cells include specialized immune cells such as plasma cells, neutrophils, eosinophils, basophils, and monocytes. So we have brought in an expert on the subject to talk a little bit about this hypodermis that we're going to see here at the deeper layer of the skin.
So if you look at the bottom of the slide, we can see the hypodermis, also called the subcutaneous tissue. This area is home to adipose tissue, which for the purposes of this class is categorized as a specialized connective tissue type. The nuclei of these adipose cells are pushed to the side by large, lightly stained adipose vacuoles.
Thank you. That was Cameron Brawley, our resident hypodermis expert. We're now looking at slide 96, which is lung tissue stained with H&E. At this point, don't worry about identifying specific structures in this slide. we're looking at the inside of a lung, which is primarily filled with air. Please don't confuse lung tissue with adipose tissue. These thin, scattered lines are primarily cross-sections of small air sacs called alveoli, where the gas exchange takes place. The one thing you should try to identify today are the alveolar macrophages that are generally located in the spaces between the epithelium of the lung. A lot of times these macrophages can be identified by the black bits of carbon that were inhaled from the atmosphere. These alveolar macrophages, which are also called dust cells, are a component of the innate or non-specific immune system. They can be hard to identify, but know that any structure stained black in the lung is likely a piece of phagocytosed carbon inside of an alveolar macrophage. Lastly, you might not see it in lab, but know that sometimes these macrophages can combine together and fuse to become a giant multinucleated cell that can be used to phagocytose large foreign bodies and to fight infection, for example, pneumonia. Now we're looking at slide 198, which is mammary gland stained with H&E. Here we're looking at a fully developed mammary gland. The intralobular connective tissue, which is located here within the lobules of the mammary gland, is composed of loose connective tissue. It contains large numbers of cells such as fibroblasts, lymphocytes, and plasma cells. Between the lobules, the interlobular connective tissue, seen here, is composed of dense, irregular connective tissue. Make sure you're able to identify plasma cells, one of which I've circled here in light blue. These are easily characterized by their clock face nuclei. And then down here at the bottom, I've circled an example of a mast cell, which is seen here within the dense, irregular interlobular connective tissue of the mammary gland. Mast cells are resident granulocytes, which kind of look like a fried egg. The H&E stain used here makes the mast cell granules look kind of reddish. Mast cells respond to antigen by releasing their granules and prompting an inflammatory response. We're now looking at slide 222, which is an aorta stained with Orsian fuchsin. This is a cross-section of the aorta, stained with Orsian fuchsin to reveal the numerous dark, wavy, elastic fibers that are located throughout the walls of the aorta. Note that if this specimen was stained only with H&E, you wouldn't be able to see these elastic fibers. To help orient yourself, there are 3 layers to every blood vessel in the human body: the tunica intima, the tunica media, and the tunica adventitia, or tunica externa. The tunica adventitia is the lightly stained outer layer, composed mostly of collagen fibers. The tunica media is the muscular layer, composed primarily of smooth muscles and elastic fiber layers. And the tunica intima is the inner layer, or the layer that touches the blood, composed primarily of a layer of simple squamous endothelial cells. If you zoom in, you can see that the tunica media is composed of densely packed elastic fibers, called plates, and the tunica adventitia is composed of more clearly delineated elastic fibers. I've highlighted an individual elastic fiber here in pink. The aorta is a large elastic artery that directly receives blood from the heart, so it's got to be able to accommodate high blood pressures and maintain adequate diastolic pressure. Another way of saying this is that when the heart contracts and blood ejects into the aorta, this blood is under high pressure and exerts a high transmural pressure on the walls of the aorta. Then when the heart relaxes, the aortic elastic tissue shortens and maintains the diastolic pressure. Note that elastic fibers are not a type of collagen. We're now looking at slide 229, which is a lymph node stained with silver. And we're very lucky to have our lymphatics expert here to help talk about the reticular fibers that are located throughout the lymph node.
Thanks, Taylor. This slide of a lymph node is stained with silver to help elucidate the reticular fibers that are seen throughout. Reticular fibers are composed of type 3 collagen and look a little bit like elastic fibers. And as I zoom in, you can see that these fibers are thinner, shorter, and more branched. These fibers are flexible but are not elastic and serve to help certain organs maintain their shape and structure. In other words, reticular fibers form the stroma to help support the parenchymal cells. The functional cells of an organ are called parenchymal cells. Reticular fibers are found in tissues throughout the body. However, you can't see them histologically unless you stain for them. As a clinical side note, Ehlers-Danlos syndrome, which is characterized by extremely mobile joints and stretchy skin, can be caused by decreased reticular fiber synthesis.
Thank you. That was our lymphatics expert, Amber Collier. We're now looking at slide 121, which is the ileum stained with H&E. The ileum is the terminal portion of the small intestine, and its surface is lined to help maximize nutrient absorption. From a 1x zoom, you can easily see these large foldings of the intestinal epithelium called plicae circularis. If you zoom into 4x, you can see that the surface area is further maximized by these villi lined with enterocytes. If you zoom in all the way to 20x, you can see the brush border on the surface of the enterocytes, composed of microvilli, which have a core of actin. Here you can get a comprehensive view of the intestinal villus. As I said before, the villus is lined with enterocytes that maximize nutrient absorption. These lightly stained cells, called goblet cells, will secrete mucus into the intestinal epithelial lumen. Now take a look at the core of the villus. This area, also called the lamina propria, is composed of loose or areolar connective tissue. All these little round things are lymphocyte nuclei, which function to help fight infection of the intestinal epithelium. This is our last slide today, slide 216, which is tendon and muscle stained with H&E. Here you can see a slide of muscle with its tendinous attachment down the middle, surrounded by muscle on either side. Tendon is an example of dense, regular connective tissue, as the bundles of collagen are arranged in parallel bundles to help provide its unidirectional tensile property. In histological preparations, tendons will be wavy and they'll have fibroblasts interspersed among them. We will be looking at muscle tissue later on this semester. Before we finish, I'd like to encourage you to do a couple things. You should make a list of the stains we've talked about this semester and try to figure out what they stain. So far we've discussed H&E, which stains acidophilic and basophilic structures, Orsi fuchsin, which stains elastic fibers, and silver, which stains reticular fibers. This list will lengthen as we go on, so try to keep track of what we're talking about so far. Secondly, if you haven't already done so, try to look at the blue box slides, as these slides will give you an alternative look at structures. The idea isn't to memorize slides, but instead to understand how the function of a tissue is influenced by its histological structure. Well, that's a wrap for episode 3. I'd encourage you to tune in next time for an absolutely thrilling look at bone and cartilage. Thanks for watching Histo Helpers. We're here to help.